Welcome to the November meeting of the Big Hole Watershed Committee. It's gonna be a little different tonight. Our executive director Pedro is in Brazil, but he's joining us remotely, so he's here on Zoom. And then I also wanted to introduce everybody to Kim Giannoni with Zoom Western. She's gonna be helping us out over the next couple of months with our monthly meetings, because I will be on maternity leave in February and may or may not make it for the March meeting, so making sure everything runs smoothly for us. So the first one up is Caitlin Boren, or, oh, no, introduction, sorry. That's all right. Okay, so introduction. So I'm Caitlin Lynch. I'm the Associate Director of the Big Hole Watershed Committee. And we'll start with the
Betty Bowler viewed.
Tom Bowler viewed. Jim Griffin viewed. The Morris.
Jenna Derman, Montana Bureau of Mines and Geology.
Jim Olson with Fish, Wildlife and Parks.
Jesse Newby with Fish, Wildlife and Parks.
Dave Ashcraft, Fundraisers.
Vanna Boccadori, Fish, Wildlife and Parks.
Big Lots, Fish, Wildlife and Parks.
Caitlin Floyd, Fish, Wildlife and Parks. Caitlin Boren, DNRC.
And Giannoni, University of Montana Western.
We're just here to, hang out.
Any students or?
We're students at Newby.
Oh, okay, cool. Thanks for joining us. All right. So now we'll have Caitlin with our stream.
Like Tana said, my name is Caitlin Boren. I work for Montana DNRC. I'm the Upper Big Hole River hydrologist. I usually give a monthly stream flow report to the committee. As always, this is our Stream and Gauge Explorer for DNRC. This is our real-time and seasonal stream gauge sites. Current stream flow, all the sites are in ice right now throughout the drought management plan section. This is the gauge at Wisdom. We kind of had a weird October into November where it would warm up and cool off a lot. And I just wanted to show you guys, the USGS put the gauge in ice a couple times thinking that it would continue to be frozen, but it kept warming up. The blue, more smooth line is temperature. And then the more angular is the hydrograph. Snow water equivalent for water year 2025 is 68% of median values right now. We're sitting about right here. These are the SNOTEL sites that we use to, or that NRCS uses to calculate SWE. This is the Hypsomi SWE for the big hole. So this just means where is the snowpack relative to elevation. And they use the SNOTEL sites. But it's saying right now that we're at 113% of normal, and most of the snowpack is right around like 7,000 to 8,000 feet in elevation, which is really good. And I think that's because we had a couple of storms in the last few weeks. Precipitation, 60% of median values. These are the SNOTEL sites that they use to calculate this value. This is the standard precipitation index map from Montana Drought. And I decided to do a water year map. And so here's the Big Hole Watershed right here. And so you can kind of see that we're sitting in like D0 to D1 values for drought.
So the colors, basically, the more red it is, the more droughty it is?
Correct. And the drought team produces this map. They update it constantly every week. And it's based on a bunch of different values. So D0 is abnormally dry, and then D4 is severe drought. Climate outlook, we're still in a La Nina watch. We haven't gone into full La Nina conditions. They're predicting that it's gonna happen somewhere before December. It's a 57% chance of happening. I know.
It's almost 50.
If we do go into La Nina, they're saying that it's gonna be weak and a shorter duration. And so what that means is that it's going to be less likely to affect weather conditions, but it still could influence how they forecast weather. So it could happen, it could not, it could influence it, it could not, basically. Oh, the 8 to 14 day temperature outlook is below average. Precipitation is above average. And then seasonal, which is November, December, January, they're saying there's an equal chance of it being higher, above or below average. And then they're saying that the precipitation could be above average. This is the week of 11/12 or of this, excuse me, November 12th drought map currently. So this is just a more detailed map that they produce. So right now we're in extreme drought conditions D3. Okay, that's all I got. You guys have any questions? Thanks.
All right, we switching to me, Tana? All right. You guys hear me okay?
Yeah.
All right. Greetings, everybody. Sorry I can't be in the room. There's been a lot going on. Run through a list of all that's happening from afar. It's amazing how busy a guy can stay out here. I'll start kind of in the beaver world, the Montana Beaver Working Group that I play a prominent role in. We got together, I lead the regulation and permitting side of the group, and we got together just a few weeks ago, looking at a couple things, ways that we can improve permitting and regulatory front and in relation to beaver and beaver management. A couple things came out of that. For a long time, I've been talking about some changes to the Army Corps permitting involved with low-tech kind of beaver-related restoration. So we came up with, there's about 5 language changes that have been proposed, and most of those looks like it's moving through the Army Corps' process at the national level. And the most important of those being that it's looking like for the new nationwide 27 permit, what is going to be proposed is that you're not going to have to do a wetland delineation to do a beaver-related restoration. I mean, the big news about that is it saves us about $10,000 a mile to do these very simple projects. You don't have to calculate the amount of wetlands existing in an area that you're going to improve the wetland condition. So, I mean, it seems obvious, but it just wasn't a part of the rulemaking. So along with that, we received FWP put together, they got an RFP out with the Utah State folks, and they actually did a model. Well, they actually looked at, for the entire state of Montana, where there's actual beaver presence using aerial imagery. So I actually got a data layer just for the big hole of where all the beaver are. I can't share it quite yet. It's still in the works, but I think it's a pretty important data set to have, and it'll help us with kind of our watershed planning that we're going to be doing, that I'm going to be doing a lot of in this year to come. Along with that, the Broadreach Fund, they gave us some funding this year to advance kind of beaver related restoration they invited us to reach out again and put together another proposal for them in the coming weeks to continue. That'll keep funding my participation in this beaver working group, along with a couple projects that we've got in the works related to potential translocation of beaver and an EA that is being finalized. And hopefully by next year, we will have a process by which we can take nuisance beaver away from areas where they are, not being helpful and being a burden to irrigators, and put them places where they can be helpful for us in storing water where they're out of the way. And with that, I'll also be participating in a beaver kind of fish work group, just practitioners around the state that are working on this interface between beaver and fish. Obviously, we know in the Big Hole, not all beaver dams are great. A lot of times they prevent fish from moving from the main stem up into cold water tributaries. So, just the science on that is always evolving, and I'm going to be kind of in that group and learning a lot. So that'll be exciting. And this Broad Reach Fund proposal will help keep me funded to stay engaged with that group. On the project front, there's a lot still going on. We've got some RFPs that are hit the street. I'm getting some bids back on this fish barrier for Rock Creek. That is set to, I think actually today was the due date for that one. I have at least one bid in for that one. In the Mount Hagen area, we have this demo area that we've been working on for years, and it needs a heavier touch. And we have some design money already allocated for that. So I put out an RFP to get some design work done there, and I should be getting those back soon. Next on the list, Smith Sage Springs. We did the restoration project in the wetland, and now the next phase of that is that entire Smith Springs system is going to get reconnected to the North Fork of the Big Hole River. And to do that, the landowner has agreed to let us siphon his ditch basically under that whole system, which is really exciting. We did a really awesome restoration project there, and now that whole system will be free flowing and reconnected. So the first things first is we need design money. Well, we have the design money. Cass and U.S. Fish and Wildlife Service had secured that already for us. So we're doing the administrative part of getting that contracted and going. And then we have high meadow storage. This is kind of a GIS exercise that we got a grant through with the Anaconda Deer Lodge Conservation District to get a GIS firm to look at the potential storage that we can get in our high meadow areas using some basic mapping tools and elevation models. The alternative to one reservoir is thousands of smaller reservoirs. So that's something that we're going to be exploring with that funding. The images you see on my slide here, these are from this Big Hole River Watershed Monitoring Report that we just finished up, and this was funded by Future Fisheries. We basically went back to Future Fisheries projects that had been funded in the Big Hole that nobody had gone back to in over 5 years. So we did 10 of these projects last year and went back and revisited about 11 of them this year. Our intern, Emma, was critical in doing that work, and she did the bulk of this report, and I just kind of wrapped it up and finished it for her, turned that in. So that was really fun to see. And, by and large, a lot of these projects are still, functioning, and the landowners, by and large, were really happy with them. I think there's some agreements on maintenance of fences and things like that didn't go as planned, but by and large, these were super successful projects, and it was fun to go back to them. With Mount Hagen, I owe Vanna a scope of work for the 2025 calendar year and a summary of what we did this year. It's still coming, Vanna. It's on the list. There's no shortage of vitamin D, but I'm also probably the only one sweating right now. And, I just found out 2 days ago that the RDG program funding list came out, and our request for funding for that remediation project got ranked 6th out of the whole lot, which is pretty good. So $300,000 is an awesome down payment for that remediation. The Forest Service is going to have to do a bulk of that funding for us, but this, is a good start. Let's see, on the people front, thank you, Kim, for stepping up and jumping into the fray here and helping us keep our meetings going. That's going to be super helpful. It's great to have you on board. I found out that the Beaverhead Watershed Committee is going to go through a change here. Zach, who's been with them and the Conservation District for many years, is going to move on to the Montana Land Reliance starting in January, I believe, of this year. So that's going to be a big change. We have existing grants with them for Elkhorn. They've been great partners. So just looking forward to, working with whoever ends up replacing Zach. Those will be big shoes to fill. On irrigation projects, I've really struggled to get funding for some of our irrigation work. And when I presented to the Bureau of Mines, I ended up sitting next to a gal from the Farmers Conservation Alliance, which is a group I hadn't heard of, but their expertise is on irrigation modernization and plans. They're out of Hood River, Oregon, but work in Montana, Oregon, and Idaho. So I had a good chat with them and their executive director, and really going to put together kind of an asset list of sort of what we know about the irrigation systems. We've done a lot. The Watershed Committee for years has characterized and prioritized our irrigation infrastructure, sort of what the needs are. So we are going to just kind of put all that information together and send it their way and see what they think. They might be able to lend a hand, and they seem to be real good at getting funding for irrigation projects. So we've got a couple that didn't get funded in the Wise River and Jerry Creek, and we've got a couple other projects that need funding in the pipeline. So I'm hoping that'll be super helpful. It's all about partnerships and getting other people around to help us out where we're struggling. And then lastly, we do have some money from the High Stakes Foundation to kind of boost our communications. And so I'm still working on what this really looks like, but we'll be putting it out in the streets, I think, early next year to get a contractor to do some science communications and content creation for us. I'm open to ideas on what that would look like. But I think there's a lot of great science and information generated from all of these meetings. And I think we can do a lot more with the content that comes out of these meetings and presentations and PowerPoints and get it out to the broader public. So that's what that's all about. And let's see, on the sort of more boring administrative side of things, all our bills are paid. That always feels good. And our newsletter and annual appeal are coming out soon. So look for those in your mailboxes. I've just got one final round of edits, and we'll be shipping those out on the newsletters. And then the annual appeal, I'll be writing them from here and mailing them to Tana, and then she'll get them out to folks by the end of the year. That's all I got.
Any questions for Pedro? Directors report, or I mean, steering committee, right? So, Roy, I think that's you in there.
Yes. Yeah. It's gonna be the shortest one of the. We didn't have any-
It'll be pretty short too. I just on events, so I staffed a table at Patagonia on October 24th for an event that they had and talked to quite a few people. And then upcoming on December 3rd, I'll be speaking in the University of Montana Western Community Water Conservation Panel there at the college. along with Caitlin and Caitlin, they're also speakers. And a couple other, people, so. That'll be fun, it's from 4 to 6 p.m. if anybody wants to attend that.
There'll be refreshments also. And you guys should go anyway, because these meetings are great. They're like put together to make sure that the community is present and has a voice. So you get to listen to Tana and Caitlin and some other folks, but you also can be really present and like put your concerns out and also just ask questions. It's good, and the snacks are usually good.
There, I've been doing a couple of them before. Really stuff. Alright. And then just a couple of publications that have come out recently. Well, this one was back in May, but it's all about our committee and our work, so it's pretty good. And then Pedro was interviewed by the Montana Cayman on the fate of the Arctic railing. So both of those we've posted on our social media, or if you'd like, you could reach out to me and I'll send it there too. And like Pedro said, our fall newsletter is coming very soon. Just a quick wildlife update on the funding side of things. So the Heart of the Rockies initiative and Montana Fish, Wildlife and Parks have secured a bunch of funding. It's like $12 million in funding from the NRCS and the National Fish and Wildlife Foundation. The NRCS one is called the Regional Conservation Partnership Program. It's 5-year funding. They have a bunch of money. In 2025, starting in 2025, they want to fund range riding, and then starting in 2026, they'll fund carcass removal. But they want to contract directly with producers. So that doesn't help us that much. There's some potential if a producer wanted to enter an agreement with us and sign over a portion of their payment in exchange for us doing range riding or carcass removal. It's a possibility. But the producers we've talked to don't seem that interested in signing an NRCS contract. So that being said, if anyone is interested in signing up or learning more about that, you can reach out to me and I will get you all the information you need. They have, like I said, they have a ton of money, so, and it's based on how many animal units you have. So it could potentially be a good thing, and even if you just wanna, if you're doing range riding and you just wanna keep the money, I can get you set up with who you need to know to get that done. On the other side of things, the National Fish and Wildlife Foundation grant is called the America the Beautiful Challenge Grant, and it's a 4-year grant. And that one's just a little more straightforward because they will sub-award to community-led groups like ours. And so that's the one that we'll probably be going after to fund our wildlife programs for the next few years. And so you can only have one or the other. You can't get RCPP and the NIFWIF grant for the same program, so you gotta choose. So anyway, but it is exciting because there's a lot of funding that's gonna be available. And that's all I've got. We're kind of in the off season for our wildlife programs right now. We're just giving Chet everything he needs for next year and looking into trying to get a 4-wheeler for him so he doesn't have to keep just destroying his. And then, yeah, we're gonna do carcass removal again in the spring, so. That's it for me, and then. Let's see, the next thing on the agenda is new business. Is there any new business that anybody wants to discuss? I should have said, does anybody have any questions on the wildlife stuff?
Yeah, with the funding for range riding, what kind of requirements do they, like stipulations do they put on the range riding? You know, what kind of oversight does it have?
That is a good question. I don't think there's much for oversight. I don't know that much about the stipulations. That's a good question. I don't really know off the top of my head, but I could get more information about it. All right, so if there's no new business, we're just cruising along here, but if there's no new business, we can do a break for 10-15 minutes and have some coffee and cookies, and then we'll come back and hear from Jesse and Vanna.
Well, actually, can I say something real quick?
Yeah, go ahead.
So a lot of there are actually quite a few people on this steering committee, they're not here tonight, that I did some research last year, talking to ranchers, talking to producers about irrigation and drought management. And my abstract was actually accepted into the American Association of Geographers Conference. So I have the ability to go and talk to a bunch of people who know nothing about ranching, and to explain the sustainability practices that you guys are using, to also explain some of the serious issues you're facing as producers, such as single species management, urban development, loss of transgenerational range workers, increasing economy problems, things like that. So if anyone has any other concerns that want me to sort of bring up during that, it's an audience of people who make decisions and vote that don't live here. So if you do come up with anything that you feel is pressing, I can probably include it. But I really wanted to thank the people that participated and just to thank our community, because I know that you're probably tired of listening to people asking you questions about what you're doing. But if I can kind of get your voice out there, I just think it's a little bit of an important thing. So if anybody has any kind of concerns or questions, it would be cool. I can like bring that up. So thanks.
All right, we'll take a break now for a few minutes.
Folks, if you want to get started. All right, thanks, everybody. I'm Vanna. I think I know most of you, wildlife biologist for Fish, Wildlife and Parks based in Butte. So I know in years past, we would come here and we'd give you trend information on our big game. But I think last year, maybe we started just giving you highlights of some of the projects that we've been working on, which might be of more interest to most folks anyway. So I'm gonna touch on 3 or 4 different projects. Not all of them are mine, some I'm just going to give you presentation on, but this one I'll start with is the Highlands Bighorn Sheep Project. And most of you probably have heard me stand up here before and talk about the Highlands Project. So basically, the Highlands Bighorn sheep, they had a die-off from pneumonia back in the 90s. And there was probably 300 to 400 sheep at the time. And that die-off took out greater than 80% of the herd, so left less than 100 sheep. And then over the years, we did a number of transplants trying to get the herd size back up, not necessarily 300, but just to get it back above 100 and get it proliferating again. And we kept getting, the population just stayed depressed. And over that time, there's been a lot of information, a lot of studies done on bighorn sheep and pneumonia across the western states and provincial western provinces in Canada as well. And so one of the technologies that people are trying now, or one of the methods, is something called test and remove. And so that's what we have been trying here in the Highlands. And basically, the testing part is you test a good sample of the sheep, you identify who are your chronic carriers of the pneumonia, and then you remove them from the population and see if that equates to better herd performance. So that's the overall what we've been doing here in the Highlands. And I just want to give an acknowledgement to Kelly Prophet is research biologist for Fish, Wildlife and Parks. Dan Walsh is a research biologist with the Wildlife Co-op unit out of University of Montana. And then there's a graduate student, Caitlin Vega, who's on the project. And so those are the 3 brain power behind the project. Okay. Okay, so I kind of just summed this up, but so the problem is we have this chronic pneumonia, you know, it's been 30 years, it's persisted. We just can't seem to get the sheep population, you know, we're sitting at about 150 sheep, and we always seem to have pneumonia in the herd that keeps it depressed. So the study, as I said, we're trying out this test and removal component. There are other states that have been trying this and they've been having some pretty decent success with it. I wanted just a little educational component here. Mycoplasma pneumoniae. Over the years, the past 30 years, we've learned that it's with this pneumonia, there's most of the time, like 99% of the time, associated with sheep that have pneumonia, you find this Mycoplasma pneumoniae bacteria. And that bacteria generally gets on the cilia of the lungs, and it renders the lungs' inability to kind of function properly. And so it compromises the lung function, which then predisposes that animal to pneumonia. So the common thing now is to test for this Mycoplasma pneumoniae. If you hear me say MOV, that's just the shortened version of it. So what we're hoping to do is identify who are our chronic shedders of pneumonia that have this MOV, remove them from the herd, and the outcome that we're looking for is increased lamb survival. And then also improved herd performance, that the herd health overall has a low prevalence of that MOVI if we could get rid of those chronic shedders. And then we should see a larger population growth than what we've been seeing. And the lambs, they are the most susceptible, of course, because if there is pneumonia in the herd and this MOVI, the lambs are fine once they are still nursing and they're getting the antibodies from their mothers. But once they get weaned, then that's when we start to see a lot of lamb mortality because they don't have the resistance. And so we have been doing an active lamb capture event and going in there, putting little collars on those lambs and then tracking their mortality or their survival. And in a lot of cases, especially this year, a lot of the cause of mortality was from pneumonia. Last year, we didn't see as much lamb mortality caused by pneumonia. And I suspect part of the difference is a wet summer versus dry summer. A dry summer like this year, there's a lot of dust in the air. A lot of these lambs, a lot of these herds are by the roads, Maiden Rock and down at Notch Bottom. And so they're hanging out by the roads, people are driving on them, the fishermen, you know, people just going back and forth. A lot of dust, and if your lungs are compromised, that dust is probably gonna exacerbate that, so. That's just my theory, haven't tested it. Okay, so part of this study is to put GPS collars on the sheep. And then to monitor their movements. One of the things that we've been able to do is to identify that there are really 5 subherds in the Highlands Bighorn sheep population. They don't function as one big herd that mixes and mingles. There really are 5 subherds. And they're shown by the different colored polygons here. This one that has 2 red polygons, this is what we call the Red Mountain herd. And the reason that there are 2 polygons is because this is the only herd that we have that actually is a migratory bunch. They will winter down here in the Camp Creek area, and then in the summer and the lambing season, they go up into the highlands. And the capture crew has been really great. The lamb capture crew, I mean, a lot of them have been trudging through snow in May up in the highlands, East Peak, Table Mountain, trying to get to those sheep because they will go all the way up there to lamb, and then they spend their summer up on Red Mountain and Table Mountain. So, but the rest of these herds, this is over at Maiden Rock. We have this Sheep Mountain herd, we call it. So Icorn's Field is right here, and that ridge that you'll see, well, that way. And then they will cross the big hole over there by like the bridge and the blue pool there. And then the marsh herd, and this herd pretty much is right across the river here at the Silver Bow Club, and then they'll go up into Cattle Gulch country. The Foothills herd just kind of works back and forth between the Guilfoys and Moose Creek. And then the Notch Bottom herd, they'll be up on Bell Peak and then down, oftentimes you'll see them at that Cottonwood Ranch. And I guess there's about 37 of them there right now, 5 rams. It's breeding season. It's a good show if you want to go sit and watch. And sometimes they will cross the river and go to the Hog's Back as well. So here's just kind of a snapshot of, there's a lot of information, but by each of the subherds, estimated group size, and then how many sheep that we've collared in those herds. And this is the 3rd year now that we're doing it. So in the Foothills herd, we have 17 adults collared. And these are cumulative. So like where we have 10 neonates, which are your newborn lambs, that's over the past 3 years. It's not necessarily that we have 10 on the air right now. And then is every time we get our hands on a yearling or older sheep, we'll take samples from them. And so then this bullet is the number of sheep that we've been able to grab samples from. So adult females, 13 adult males sampled in that herd. So for each one, the group size, and then the number of animals that we've actually had our hands on, put collars on, and the numbers that we've been able to get samples from. And in most cases, most of these are multiple samples. Because that's the whole idea, is we have to capture as many as we can each year, as many of the same ones each year, so that we can identify what we are calling a chronic shedder. And that's an animal that is testing positive for this mycoplasma open pneumonia 2 years in a row. So you'll go to the next slide. Okay, so again, little educational moment here. So this Newby predisposes the animal to pneumonia. We're doing 2 different tests when we get the animals in our hands. We're doing a serum test, it's called the ELISA, it's a blood test. And that gives us an idea if they have the antibodies in their serum. And that tells us that they've been exposed to it. It doesn't necessarily mean that they have pneumonia, but they've been exposed to it because they have the antibodies in their blood. Then we'll also do nasal and tonsil swabs for the bacteria for this mycoplasma pneumoniae. And if that comes back positive, that means they're actively infected. So we're looking for individuals that are showing an active infection 2 years in a row. And we're calling those our super shedders or chronic shedders, typhoid Marys, whatever you want. They go on our hit list. So, but I want to just point out, so this is kind of a busy graph, but I highlighted Sheep Mountain. So these are all the different subherds, and these are the prevalence. This is again your serum. So this is that they've been exposed to it. This is the PCR. This says they are actively shedding it. But it changes from year to year. That's really what I want to point out to you here. So in 2022, in the Sheep Mountain herd, we tested 13 animals. And we had prevalence 0.7, so 0.7 of those, or 70% of those 13 had exposure to it, but only one of them actually was chronic at that time, or not chronic, I'm sorry, actively infected. In 2023, we tested 16, many of those were the same animals. And the number that were exposed to it went down, but the number that were actively shedding went up. And then in 2024, we tested 11 from the same herd. And all of a sudden, just about all of them, 80% of them were exposed, had been exposed to the pneumonia. But the number that were actively shedding went down. If it makes sense to me, if you had this many actively shedding, then the next year you're gonna have a lot more that are showing exposure to it. But I wanted to just point out the dynamic nature of this. And that's why we can't just remove those that are testing positive once, because they will clear it. Some of them will clear it. Even some of the lambs will clear that pneumonia. But the ones that don't clear it, those are the ones that we will. So, and one other thing I want to show you is that in a lot of cases, we will, like everything, you have, your one general MOV, but then there are several strains of it. And so in some cases, we have sent it to the lab to get strain typing done on it. And most of the sheep in Montana, when we have done strain typing on the MOV that they have, it comes back to this BHS 29, which is associated with the Yellowstone herd. And a lot of our sheep have that strain, but we also detected a strain that has not previously been known in Montana. We've had some sheep, some of our sheep in the Highlands come with that strain. So not sure exactly what that means, but that's all part of what's on the table to figure out. So. Okay, so our basic time frame for the study is that it's a 5-year study, which is really nice to get that much time. Typically, these studies are 2 years, and you get a quick snapshot and not always an accurate one. So years 1, 2, and 3, we've been capturing the sheep, putting collars on them, and testing them. And then we are focusing on adult ewes and yearlings and a smaller number of lambs. Those adult ewes that are collared in the springtime, we also have put bits in them, which are vaginal implant transmitters. they get expelled when a birthing event occurs, so then we know that they have just lambed, and so then our crews go in there within, oftentimes within a day, you know, we tell them to wait 12, 16 hours so that that lamb and mother can pair up. But then they'll go in and they'll just quickly gather some information, put a collar on that lamb and release it, so then we have those lambs collared and we can track their survival as well, so. So we are right now in year 3. In years 3 and 4, we are removing the chronic shedders. Year 5, we're monitoring for those metrics, the lamb survival and the herd health. So we have sampled enough animals and multiple times that we identified 7 chronic shedders this year. 3 of them died on their own, just various causes. And so back in September, we removed the other 4, and we just took them out with gunshot and took them out of her. So we'll start here right after the first of the year with our winter capture. Most of it's gonna be helicopter capture. We might try to do some drop net as well. But we'll just do our next round of making sure we have a lot of collars up there, to get more sheep sampled, identified, chronic shedders, and those will be removed next year, so. So that's where we're at with that. So any questions on the Bighorn Sheep project?
What do you do with the dead ewes?
The ones that we removed?
Yeah,
Yeah, so we talked about like donating them to the food bank, but it's always a little tricky conversation because the reason we took them out is because they have a disease. And then we're giving them to the food bank. So it's always a little, yeah. We did give some meat to one of the landowners who has been very cooperative. So it's available if somebody wants some sheep food. Just know that we took them out, yeah.
but I guess just, yeah, carcass removal, you know.
Oh, so, yes,
I'm curious
they
what
go to
actually
the
happens.
lab,
Okay,
full meat crops.
okay.
I know where you're going. Okay, all right, I'm always thinking in terms of meat. But, all right, gotcha. Okay, so any other questions on the Bighorn Sheep project? All right, so I just want to give you a CWD update. This is based on the report that just came out from our folks in the lab and the folks that are heading up the CWD program for the state. So this is all 2023, 2024, not including this one. So, but during that time frame, we tested over 7,000 samples. Most of them came from mule deer and white-tailed deer, some from elk, some from moose. Of those, we've had 238 animals test positive, 86 from mule deer, 151 white-tailed deer. We did have 1 elk test positive. And again, this was from this past year. So we did have CWD crop up in 3 new districts in the state last year that we didn't have detected before. 213 is probably the closest one to home. It was a deer that was, I believe it was a mule deer from the prison ranch over in Deer Lodge. So a couple of things, I just wanted to, like, the highlights from this whole study, and I did bring a copy of the study if anybody wants to look at it, but close to home in hunting district 322, that Ruby herd, basically 1 in every 3 deer test positive for CWD over there, white-tailed deer. And then we did have some positive show up in the Highlands. 13% of the tests in the Highlands came back positive. All of those were in that Twin Bridges. I think Jim contributed most of the samples. Wait, if we go back. Yeah, so if we have not found it in 340, we haven't found it any further upriver past Twin Bridges. So I just want to point that out. And then we have seen some patterns in the prevalence, and there's been some literature leading up to this, but now Montana has been, that we use that literature from other states to kind of inform our management plan, but now we've been sampling long enough that we're starting to see some patterns here in Montana, and it just kind of strengthens these other studies that were done too. But basically, whether you are a mule deer or a whitetail, if you're a buck, you have equal chance of having CWD. If you are a female white-tailed deer, you are 3 times as likely to have CWD than a female mule deer, and these are adults. And then If you are a male deer, have a slightly higher chance of having it than a female deer. So basically, adults are most likely to have it, yearlings followed by that, young of the year. Probably not going to have it, but they're less at risk, I should say. And then this is just a map of our areas that we prioritize for CWD samples in 2023. And then we have 2 CWD management zones, the Libby area and the Ruby area, where we've been really trying to target harvests in those areas. Okay,
If somebody harvests a deer or an elk and it tests positive for CWD, do they get a new tag?
right? Jim, you got it?
Yeah, unless it's a buck, you have to trim your horns in if you want that.
Yeah.
Can't keep the horns.
Yeah, and that kind of follows with any time, if you have an animal that is deemed unfit to eat, you can get a new tag, but you've got to turn everything over, including the antlers, and sometimes people are, well, it's my son's first year, so, you know, they keep it, but they don't eat the meat, but. Oh, let's go back for a minute. Okay, just want to point out one other thing. Some of you may have seen in the Montana Standard about 3 weeks ago, there was a good article about the health lab over in the Bitterroot in Hamilton, and they have been doing some studies on CWD, trying to get it to cross the species barrier to humans, using human cells in the lab, not real people. Yeah, if you want to volunteer. And they have not been successful in getting it to cross that barrier. And there is some potential that it's a different strain, it's those strain things again, so. Anyway, it's not the end all be all to the research, but preliminarily, their rigorous testing in the lab is not to get it to cross the barrier. So, okay. Yeah,
Before we leave that, was there any theory as to why white-tailed does are so much more high risk than mule deer? Is it like a behavioral thing or how do you?
this is my guess, is that partly it might be an artifact of our sampling and the targeting of the ruby and whatnot, and where to have it, so, yeah. I think if we just did a blind sampling, it might look different. That's just my guess. Okay. Any other questions on CWD? All right, wolf management, again, just kind of real quick and a snapshot based on the 2023 report. So this doesn't have 2024 numbers necessarily. Population estimate, I'll just get right to it. For 23, estimated just over 1,000, well, 1,100 wolves, and you know, confidence interval going from just under 1,000 to 1,200. We use something called IPOM, which is the patch occupancy model. So we basically are asking hunters at the end of this hunting season, when we call off about elk tags and deer tags, and we're asking them, did you see any wolves? And so we get all that information. Did you see wolves? We get an idea of what the distribution was for the past year. And then on top of that, we overlay territory sizes and pack sizes, all based on that year's information. We just kind of put that all together and work the magic, and then it spits out this number of estimated wolf population for the state of Montana. So 1,100 wolves last year. Again, this was for 23. We had 254 wolves harvested, 144 in the spring, 110 in the fall. Wolf hunting licenses in 2023 generated $285,000. That goes back into wolf management. And then livestock loss for last year was 32 animals, mostly cattle. And then the payout from the wolf reimbursement livestock loss was about $43,000. All this is here on the graph. So the red line is our baseline. This is what we are obligated to maintain wolf populations at. If we get anywhere close to the line or drop below it, the wolves could get delisted again. The blue bars are the estimates of the wolf population in the state. So we're not anywhere near that red line, which is really good. Wolf harvest is represented by the black line. And you can see it's really, I mean, if you just kind of squinted at that, it's really been pretty steady for the past 8 years. So this is without like any real statewide cap on harvest. This is just, this tells us that, you know, we've kind of saturated the interest that's out there and the abilities to harvest. So, okay, any questions on wolves? I think that's the only slide I had about that. All right, and then I have one last one, and I was hoping Eric also would be here. Sarcocystosis. So this is something, it's a protozoan infection. It affects muscles. It looks like these little grains of rice in muscles. We have seen it in some elk that have been harvested on the west side of McCarty Mountain in that Earls Gulch area. And it basically is a 2-host life cycle. So you have to have a definitive host, and that's typically your carnivore, your scavenger, humans. And the infected host, if it has this protozoan in its system, that protozoan will kind of do its reproductive thing. And so then the definitive host will poop out what's called oocytes, kind of like eggs. The intermediate host comes along and either eats the feces or the contaminated vegetation or water around that area where the feces were and ingests it. And then those oocytes will travel through the system and they'll go through different lifestyle or life stage changes, and they'll end up finally arriving in the muscle and form these little sarcocysts. And then the scavenger or carnivore or humans come along and eat it, and the cycle goes all through, it goes through the cycle again. So basically, if you have harvested an elk, and we've had this conversation with a few people who had harvested elk in this area, and they had these sarcocysts in there. Generally, the current literature suggests that humans are the definitive host. The definitive host generally does not get sick by eating those cysts. It poops them out. It is the intermediate host that will show the symptoms. However, you know, if you don't cook your meat, I mean, the answer to a lot of this stuff is just cook your meat properly, and it will kill those cysts and render them unviable. So, but as long as we are in the definitive host, we're not likely to get super sick. If you are, like, if for some reason you came in contact with the feces or contaminated, you know, water or vegetation or whatever, you know, from that has those oocysts in there, and you ingest those, then you become the intermediate host. So if it's just the meat that we're worried about, you've got that muscle, just cook it. And I think it's like 165 is the recommended temperature. It's just like tapeworm cysts or things like that. If you cook it at the proper temperature, it will destroy those cysts. So great slide to end on. Any questions? Cookies? Yeah, those cookies probably are worse for you than the. I didn't let you take them.
I didn't.
Yeah.
So the spread of chronic wasting disease seems like it continues to spread to new areas. What's to stop that? I mean, they talk about carcasses, right?
Yeah,
Is that why it's spreading? Because
I mean, that's
animals are
one
dying?
vector.
Huh?
That's one vector. Some of it is not necessarily a spread so much as we just started looking for it there. So that's the other thing. Well, you know, like all of a sudden it seems like we got CWD everywhere, but now we're looking for it everywhere.
I see.
So, yeah, that has to get teased out. Is it really spreading, or are we just now paying attention? So,
And then on the sheep, that bacteria that's causing the pneumonia, is that all inherent to the herd, or is that coming into the herd from somewhere?
yeah, that's a good question. And, you know, I mean, there are ways that it can be brought to the herd, but where it originated, that I don't know, yeah.
In Yellowstone, well, the Yellowstone herd.
Yeah.
Was where it came from, I'm guessing.
Well, that's a strain that's been identified with the herds around the Yellowstone area, but I'm not sure like where it originated from, so, yeah.
Oh, okay.
Yeah. And it's probably something, and so with the Highlands herd, I mean, it's been 30 years, we don't have, like, we are not in an epidemic with the pneumonia there now, we were. But now we're in endemic levels, I mean, it's just like with COVID now, where the epidemic's over, but it's still out there, so, yeah. So it's not like we're gonna get rid of it, we're just gonna try to suppress it and minimize it being in the herd by getting rid of the super shedders. We'll see if it works. Yeah. Okay. Yeah, Tom.
That other strain you found, could that be a mutation that's occurring?
It could, I'm guessing because it has a number, it's known elsewhere in other herds. And wasn't just a new one. It's new to Montana, but I think it's been seen in other herds in the West, in Canada, so. Yeah. Any other questions? All right, thank you.
Okay, so I'm actually gonna talk about moose, but we'd probably be remiss not to mention some mountain lion work that's about to occur here. So as part of our statewide monitoring and management of mountain lions, we have the state broken up into different ecoregions. And then within those ecoregions, there's areas identified that are kind of representative of that region where we get actual population estimates. We do this by contracting out houndsmen to run cats and shoot them with a biopsy dart to get us a tissue sample for genetics. And so it's basically marker capture using genetics. This year and last is Southwest Montana's turn. This kind of is a roving sampling that goes around the state. Southwest Montana's turn, and this winter it's going to be in this area, so into the Pioneers, Highlands, up into the Pintlers. And so basically we have this area gridded out, randomly select cells, run cats there and do the sampling. This year will be different than previous years where this has been done because we will be collaring some cats as well. We're going to try to collar up to 10 cats to try to better inform how these marker capture models are estimating the populations. So they use things like space use, habitat use to try to refine those models. There's been some questions about how well they're performing. So having collars out there, we can actually look at the cat movements and what habitats they're using. Hopefully improve those things. So I don't know how many houndsmen are out there, but we are cognizant that we don't want to step on people, and blow their hunt. So we will try to take measures to kind of minimize our impact while we're running these cats. But you might see our trucks out there. That's pretty much it for mountain lions. I guess before I leave the mountain lion thing, I'll just mention if there's questions that are come up while I'm up here, just go ahead and shout them out. I'm happy to take them as they come. So if there's no questions about the mountain lion stuff, we'll just move on to moose. Many of you are probably familiar with the Moose Project that's been going on for 10 years. And maybe wondering why we're talking about moose again, but the reason for that is the new development is that the study's been completed. So the whole study's been completed, most of the analysis done, the final report's been completed. There's some loose ends with some manuscripts that are in prep for for some journal articles, but for the most part, we're done, and we have the results that can be shared with you. So, I guess we progress. Just a reminder, in case anybody hasn't seen this, the motivation for doing this research was concerns about declining moose populations. And you can kind of see some representatives of that in terms of the number of tags that were given out in the past, since the 90s, we've dropped tags considerably, particularly here in Region 3 and Region 1. And this kind of reflects concerns about the population. And also, it seems like hunters are having to put more work in to get moose. So this graph right down here shows the number in the solid lines, the number of days moose hunters are putting in the field. You can see that increasing over time. And then the open triangles are the kills per unit effort. So for every 100 day, how many moose are killed? And you can kind of see that declining. So it seems like hunters having a tougher time finding moose, maybe there's less moose out. And there's been similar concerns in neighboring states, kind of in the southern edge of moose range. I think we can go.
Ask, what,
Yes.
why is there such a big increase between 1950 and 1960 in moose populations?
That's tags.
Oh, it's.
So that's
oh.
tags. So they were really conservative. I think the first. They basically closed down hunting, I think, around 1900, give or take a few years. And they kept it closed till almost 1945, and then they were just really careful about doling them out.
Is that
And
next?
then they
line?
just really liberalized it.
Is that next line population?
No, none of these
Those
are
are all
populations.
tagged, okay,
Because we don't have any estimate of statewide populations
okay,
until now, but I'm not sure.
okay.
So the two goals of the research was to try to evaluate monitoring strategies for moose, because like I said, we don't have those population numbers. And then the other one was to actually drill down on our population dynamics and look at what might be limiting those populations. In the interest of time, I'm not going to talk about the monitoring stuff. I wish I could, but just to get us out of here quickly, I'll just focus on actually go back. So I'll just be talking about the population dynamic stuff. And there was 3 study areas where we drilled into those populations to look at population growth, vital rates, limiting factors, and those are up in Northwest Montana, Cabinet Salish, on the Rocky Mountain front, and of course, the big hole here. So now, move on. So that's the man himself, that's Nick DeCesar, who led the study. Looks like Pedro's stepping on my numbers there, but basically, I think I remember him anyway. So over the course of the study, we captured 193 cow moose. 186 of those had either VHF or GPS telemetry, which we tracked over time. And that comes out to about 612 moose years being followed here. And from these captures, we get things like, and the collaring, we get those vital rates like adult survival, fecundity, calf survival. And then while they're in hand, we can look at other things like nutritional condition and things like that. And of course, since we have the collars, we can also look at how they're using space and how they're using some habitats. So just on that front, looking at space use, this is estimates of home range size in winter and summer. So winter here is just looking at space use for January and February. And you can see that on average, the moose are using, or median value is about 12 kilometers, square kilometers. In summer, so this is July and August, it almost doubles. They're using kind of a larger area during the summer. And there is, depending on some of the moose, varying levels of migratory behavior. in all the study areas. And I thought you'd just kind of be interested to kind of see how the moose were using the big hole landscape. And so what this map shows is, I think it's 60 moose. And these is just kind of a representative location in summer and winter. So the yellow is the moose location on August 1st. The blue is February 1st. And so this kind of gives you an idea, like kind of how they're moving across the landscape. And you could see, I think almost 2/3 of them are actually quite happy just to stay in the Big Hole Valley proper. Then about 1/3 of them, maybe a little bit more, do head up or kind of elevational migrants going up into the forest and using the higher country. And I think we can move on. So I kind of wanted to keep this more big hole centric, but I did kind of want to mention some of the work that was looking at habitat use up in the Cabinet Salish. Because I think some of the things might be relevant to some of our moose habitat in more forested areas, let's say the pioneers or something. And so this was work, the analysis and write-up of a lot of this was by Rich Harris. And what they looked at was so this is basically looking at habitat selection and looking at these different variables like topography, vegetation cover, distance to some linear features. But the big interest to motivate a lot of it was in relation to timber harvest. So as you probably know, moose eat shrubs. And it's been thought that in especially these forested environments like that they really rely heavily on disturbance. So probably historically wildfire, maybe in more recent historic times, timber harvest to let some light in, get that early cereal stage shrub growth that they can use. So one of the thoughts is maybe we're seeing a decline in moose populations because we haven't seen as much timber harvest. Those areas are growing in and aren't as productive as they used to be. So the timber harvest was interesting. We also wanted to see their response to burns. And then look in particular where we have this disturbance, what kind of growth we get in terms of a couple of important shrub species, in this case, ceanothus and willow. And then finally, looking at how the thermal conditions might influence their movements. I think we could here. I think in the interest of time, I'll just draw your attention to this panel B. And what this is looking at is in the yellow, this is how much of this habitat type is available to them. So how much area it covers. And then the green is moose selection for that area in summer, and then moose selection in winter in blue. And so what you can see, the unharvested forest, there's plenty available. And they seem to not select heavily for that and kind of select away from it, particularly in winter. So they're using it less than you would expect if they were just kind of wandering around the landscape at random. In terms of harvest, even age stands, so these are clear cuts. We do see a bit of increased use, particularly in the winter of those areas. But where we really see the most significant difference is the uneven age harvest. So this would be like a shelter wood cut, something like that. And there we do see a significant jump in them using those areas much more than their availability, both in summer and in winter. So it does seem like they are selecting for those harvested areas. Go forward now. And the burns, so the problem with looking at burns is there were only so many moose that had burns available in their home range. So it really kind of limited the ability to look at them. But if you just look at those individuals and look at their use compared to how much burn they had available in their home range, you can see in summer, this green line shows their selection for the burn. Black line's how much is available, and you can see that they're basically selecting for it, and the more the merrier. They just keep using it more and more if they get more burn. The winter, it's a little messier at low availability, so if they just have a little bit of burn in their home range, we see selection for it. Then as you increase that amount, you see that selection disappear. Not exactly sure what to make of this, because as you can see, you know, we had a bunch of moose that just had a little bit of burn available during the winter. And then just a few moose with more, so they might be selecting for it more than this looks. This might just be dragged down by these few individuals. Okay, I think again, I'll just focus your attention on this bottom panel, this B slide. So what this was looking at was the abundance of ceanothus and willow in these different disturbance regimes. So the orange there is wildfire. And you can see both willow and ceanothus really boom in those wild. The light green is uneven age harvest. You can see willow looking better than undisturbed areas in the darker green. So a little more willow in those. And ceanothus kind of similar to untreated areas. So I guess what this says is that, you know, there is some thought like, well, historically those burns were providing this disturbance role that can just kind of be replaced by harvest. And it does seem, we do get increased shrub growth in the harvested areas. They do seem to be selecting for it, but it's not necessarily equivalent. Those wildfires appear to be providing different forage in different proportions. And then the last bit of this is looking at how they respond to the thermal landscape. So I'm probably gonna have to walk you through these graphs. Here we have canopy cover, percent canopy cover, and then down here, the time of day. And the brown dots show the relative change in temperature in these different canopy cover layers. So, for example, this will make a little more sense if I just describe it as at nighttime, those places with heavily canopy cover are warmer, right? It's insulating the ground. But in the middle of the day, they're cooler, so they're shaded. So that's what this pattern you're seeing. And you can see that the moose use just kind of mirrors it. So when those areas are relatively warm, they tend to avoid them more. And then when they cool down, they use it more. So it seems like they're really tracking, you know, these changes in temperature throughout the day. And this just shows canopy cover. If you look at topography, you see a similar pattern. So the differences between, let's say, a bottom of a draw compared to a ridge, and how that changes throughout the day, they do the same thing. So it does seem like they're making these small adjustments in how they use the landscape to stay cool. And they do it both summer and winter. I think we can go on. Okay, now I'm just going to shift over to population stuff. And this is kind of the money slide because it tells us where these populations are going. So this is showing population growth and that's based on what we see for adult female survival and then the calves recruited per female. And as you read this, basically this bold line where it says land equals 1, if you're on that line, you're stable. Below it, you're decreasing. Above it, you're increasing. And you can see that the big hole lands right at land equals 1. So stable population. Of course, there's variance in these estimates. So exactly where it's sitting is a little uncertain, but basically pure stable. In comparison, you have the cabinets and the Rocky Mountain front where you have higher adult survival. So they're performing better and a bit higher calf recruitment, particularly in the Rocky Mountain front. It makes that population really kind of booming. Now, the question is why does, why is the big hole stable and these other ones growing? What environmental factors might be acting on that? So here, population growth, that's going to be a product of these vital rates here. And they're going to influence that population growth in different ways. And those in turn can be influenced by these other factors out in the environment, things like forage conditions that will influence nutritional condition, things like parasites and disease and predation. So we kind of want to look at how each of these are kind of playing on those vital rates and what that means for the population. And so I'm going to turn to just one of those environmental factors, and that's the forage. So this is just a table of moose diet in the Big Hole. And this was obtained through collecting pellet samples in the winter and summer. And the samples, they run through a PCR to look at DNA of different species that are in the pellet samples. And then kind of look at how many those species are represented in the pellet samples to look at the proportion of those species in the diet. Some things we can't take down to the species level. So this is just willow. But you can see in the Big Hole, it's really willow dominated. They're eating primarily willow, especially in the summer. And then in winter, you get the grasses from them eating in the haystacks. Now, I'm not going to go into detail about the other study areas, but I will just kind of contrast how they look compared to the Big Hole. In the cabinets, willow is a much smaller component. I want to say it's like 14% or something, but they're really hitting our rose species. So things like chokecherry, serviceberry, thimbleberry, and cinnamon. That's another big one. In the Big Hole, they're kind of using intermediate amounts of willow, not quite as much as the Big Hole. And then other species like particularly red osier dogwood, to a lesser extent bog birch become important. But the thing that really strikes you when you look at the species that are browsed in the Big Hole compared to these other study areas is that it's really limited in the Big Hole. It's a pretty short list. If you compare it to the other study areas, it's like a third of the species that they're using in these other study areas. So much more diverse diets outside the Big Hole. And so then the question is, well, what are they getting out of what they're browsing? So to do that, Colin Peterson down there and Nick, they would go out to moose locations, see what plants were browsed, and then take clippings from them to measure things like digestible energy, available protein, tannin levels. I'm just going to talk about the energy because that's kind of the bigger one. And what you can see, if you take those plants and how much energy is available and weight it by how much that's in their diet, you can see that in the summertime, digestible energy in the Big Hole is lower than the other two study areas and significantly lower in the Rocky Mountain front, which they had much more high energy diets. And, you can also kind of look like, well, what is the minimum to keep a moose going through the summer? And they're definitely exceeding it. They're getting much more than they need. Kind of have to, because they have to wear that, right? And then the other thing that's interesting about this is they're selecting for the species that have higher energy. So more nutritional species. So they're not just kind of eating what they come across. They're picking up the more higher energy species. Now in the winter, it looks quite a bit different than that. Now, the cabinet fisher or the cabinet salish is kind of higher levels compared to the Big Hole and Rocky Mountain front. So what's interesting about this is both, especially in the Big Hole and Rocky Mountain front, they are not meeting the requirements. So they're basically living off the ground. The other thing is they're not selected for high quality species. At this point, they're just kind of putting stuff in the grass. And so really what seems to be driving things is what they can get in the summer. So the strategy, and this kind of is backed up by prior news research, seems to be in the summer, you're really seeking out energy stuff, you're really laying off that. And then in the winter, you're just hunkering down. And you're kind of in starvation mode, you kind of feel you're better. So now, going from that to parasites. So I think most of you have heard us talk about Elaeophora. So this is a roundworm where its built phase tends to be in the carotid artery of the neck. And then other stages like porphyria taking around other blood vessels. And it's been tied to a lot of these health-related mortalities for time. So we see a lot of these animals kind of tip over either from acute reasons or from chronic issues. They tended to have these higher Elaeophora. Now the problem is there's no anti-mortem test for Elaeophora. So we can't take a live animal and say, is it infected? You have to put it. And so it really makes it hard to tease out what kind of effect this is having on mortality. But Nick got pretty creative trying to tease out how this might influence health-related mortalities. And the way he did this was to take these animals that we know were killed or died from health issues compared to hunter kills. And assuming like, well, hunter-killed animals are healthy. If you do that, and so you have healthy animals versus mortalities from disease, you can see here, this is if an animal's infected with Elaeophora up here in the dark circle, and then if it's uninfected, the chances of it having a health-related mortality, and basically infected animals have about 4.5 times more likelihood of dying from health-related issues. And then the loads also influence this. So basically, this is showing how increasing intensity of infection, how many worms are in the carotid artery that we document, relates to increased chances of health-related mortality. And basically, for every 10 worms, it increases those chances by 2.5 times. So this isn't exactly a smoking gun saying Elaeophora is causing all these health-related issues, but it sure seems to be tied pretty closely. Now, the kind of last environmental factor I want to talk about is predation. So with a lot of these things, we couldn't manipulate the system experiment. We couldn't change, predator densities or anything like that. So we instead kind of had to capitalize on spatial differences in predator densities and how that might relate to what the moose vital rates were within their home ranges. And so basically what we did is we took all our moose locations and gridded out that area and randomly selected cells where we could put up cameras on travel routes to document predator use to try to get an estimate of density in these different areas. And so this is just showing, we got a lot of pictures. I mean, millions and millions of pictures. We had 90 cameras out for almost the entire length of the study. So you can imagine what kind of mind-numbing process it was to go through all these pictures. I'm just going to show you the kind of focal predator species we're interested in. And I think the one thing that obviously pops up is that, relative to the other study areas, we're getting a lot less detections. Maybe it's kind of close in black bears to the Rocky Mountain front, but nowhere near Cabot Salish, and, you know, all kind of down the line similar. Another way you could look at this is there we go. Is just the proportion of cameras where we got detections across the study area. So for example, black bears here, you know, in the big hole, we get a fair amount in the summer, not so much in the winter. But the proportion of cameras hit catfisher are certainly larger. Proportion of cameras hit in the big hole is relatively small compared to catfisher or wolves. Similar with grizzly bears and certainly mountain lions. And so I think. So that, well, actually, let me go back. So these are just kind of the raw numbers. What actually gets used in the analysis is an estimate of density that's based on looking at different habitat features, including the number of prey, for example, and how that correlates with predator detection and by extension, predator density. So these aren't actually the numbers that go into the analysis, but the model that kind of gives us predator density, or at least an estimate for it. And now, just going from the kind of environmental factors, going back to the moose and their vital rates. So these are the vital rates we're interested in. Adult survival, comes out about 612 years of moose years. Pregnancy, I want to say we had 619 in that sample, but actually might be more. And then we're looking at parturition and litter size, and then calf survival throughout the year. So there'd be different periods of the year, early summer, late summer, early winter, and then April, where we observe the cows and see if they had a calf and heel, and if that calf and heel disappeared. And that came out to about 3,700 calf conditions. So that's kind of where this vital information is from. And now these are the rates we're actually interested in. I'll just start at the right-hand side. That's the adult female survival. And you can see that the big hole is lagging behind those other study areas in terms of adult female survival. And this difference might look really small, but because adult female survival is so important to this population, this is the main thing that's been swinging, even though small differences have an effect on the population. And then, of course, hopefully those adult females have young, and so this is the fecundity rate. So this is the number of calves born per female. And you can see again, the big hole is lagging and not as fecund as particularly the Rocky Mountain front. And then we can look at calf survival at different stages and the fetal neonate period, which goes up to July 15th. And here you see really the Cabot Salish lagging. Summer, big hole calves do pretty well, but then in winter they have higher mortality. So what plays into these different vital rates? We'll start with adult survival. Well, one thing that matters is the age of the calf. So after about, when they get to 9 or 10, as they get older, the probability of them surviving drops. So this is shaping some of that survival stuff. And the other thing is the different sources of mortality we have. So all the callers had mortality signals. So we could try to respond as fast as possible once an animal died to try to figure out what happened to it, and by whatever means necessary, get a necropsy on it and determine cause of death. And this shows basically the chances within a year of an animal dying from different sources of mortality. And you can see by far and away for adult cows, the number one thing they ran a risk of was health-related issues. So that's really dominating it. A distant second is human caused mortality. So this is harvest. And the harvest is really only applicable to the big hole because we didn't have a cow season and the others. As well as vehicle strikes, fence entanglements. About half of all this mortality is from harvest. And of course, this is looking at all these study areas combined. And then third is predation. So,
What's that picture?
so that's our former pilot helping out on a moose necropsy that unfortunately the animal died in the water, so, messier. So this again draws attention to these health related issues. So these are all moose in a big hole, but they just tip over. And some of them, look like they were in bad shape, maybe dealing with kind of chronic issues, but some of them just look really acute. Like that animal looks great, and she just healed over. So this again really draws our attention to these health issues and this relationship that we see with Eleoptera. This kind of seems like, I think every cow here that we sampled for Eleoptera had worm loads and often very substantial. And this occurred in acute cases of mortality as well as chronic. So there's not really cause and effect here, but it does seem like something, there definitely seems to be a relationship.
Are you finding dead bull moose too?
Well, we don't call those moose,
Oh.
but what comes up with that and how they did enter into some of this analysis is, occasionally we'll get called in to dispatch a sick moose. And so bulls end up in those samples for sure. Now turning to fecundity, so how many calves they're having, and this is just looking at pregnancy rates. And again, you can see the age of the cow makes a big difference on the likelihood of them being pregnant. We have pretty low pregnancy rates for yearlings. Overall, it was 20 female yearlings were pregnant, or no, that's a sample of 20, about 25%. But this is pretty much all on the Rocky Mountain front. I don't think we had any yearlings breed down here. And that's probably tied to nutritional condition. And then, high pregnancy rates up here, but then after they get past 10, those rates really start to drop. And the other things that really seem to influence fecundity is nutritional condition. So this is just looking at pregnancy and some of the factors that tie into that. This first panel is if they have a calf at heel for the past year. And if they were pregnant the past year, that's these dark circles, they were a lot more likely to be pregnant this year. And what this is probably indicating is basically just individual variation. Some moose are sitting in a sweet spot and some aren't. If you're successful in being pregnant one year, you're probably going to be the next. Those age patterns are entering in that too. But what's interesting is that there does seem to be a bit of a cost of raising a calf. So this shows the probability of pregnancy in a year, depending on how long they kept their calf the year before. So basically, if you lost your calf the first week or two, you're going to have a lower chance or a better chance of becoming pregnant than if you actually put all the resources in to getting the calf off that year. And then this is rump fat. So when we capture moose, Nick would measure rump fat using ultrasound. And this just breaks it out to if they had rump fat or if they didn't. And if they had rump fat, they were almost certainly going to be pregnant. If they lacked rump fat during the winter, then the chances were not much better than 50-50. The thing that actually jumped out as the strongest predictor of pregnancy, though, was the amount of serum iron. So the amount of iron available in our blood. And this just shows the increased probability of pregnancy as you change iron levels. This dashed line shows levels considered deficient in cattle. This shows dashed line, sufficient levels of iron for cattle. And basically, if you didn't have iron levels that were better than sufficient for cattle, you probably weren't gonna be pregnant. And so this serum iron, exactly the mechanism of how it's influencing the pregnancy and how it's related to other nutritional measures is a little uncertain, but basically it seems like this is tracking its overall nutritional condition.
Are they
Yeah.
getting iron from their diet?
Yeah.
I know.
Yeah, but I don't want to get too much in the weeds, but iron uptake is a way complicated process that I had to dig in that. But it does seem like it's influenced by how much fat they have and probably their protein status is their ability to uptake it. So it's in their diet, but it's their ability to uptake it that's probably influenced. Now, this is just, I don't know how often you're gonna see a 3-point regression. But this is looking at fecundity, so calves per female. Based on that summer digestible energy in the different study areas, again, you know, it was lowest in the big hole. Indeed, we see these animals are less fecund. And, you know, significantly higher in Rocky Mountain front, where you have high fecundity, you know. With 3 points, it's kind of hard to make sweeping statements about this, but there's certainly a pattern there. And now moving on to calf mortality. So when you look at calf survival, the thing that jumped out is really the only predictor and a strong predictor of if a calf will survive its first year. So be born and make it all the way to be recruited as a yearling was the age of the females. So females, if they were 10 years or older, are a lot less likely to be able to pull out a calf. And that's really the thing that came out for annual calf survival. We can get more granular and look at different stages of that calf's growth. So for example, that fetal neonate period, so that's up to July 15th. There was a weak relationship with the combined index for black bears and wolves. And then in summer, so that's like July 15th to September 15th, the things that came out was if the cow had been successful last year in recruiting a calf, that increased the chances she'd be successful this year. Again, I think that's just variation in individuals and the places they're living. And the other thing that came out was the combined, basically all predators. So the density of big teeth out there in the landscape influenced calf survival negatively. And then in fall through winter, so this is going all the way up to April 15th, we see a weak relationship with snow water equivalents, so how deep the snow there is. Moose are built for snow, but it did seem like deeper snow levels might make it tougher for a calf to live. Again, this is a pretty weak relationship. And then predators also came out, at least black bears, mountain lions, and wolves combined. And this counterintuitively had a positive effect on survival. So the more predators you had, the more likely you're gonna have a calf survive the fall and winter. I don't know exactly what's going on there. One thought, kind of speculative, is we might be seeing some compensation. So the predators are killing off some of the doomed surplus in the summer. And if they wouldn't have, they're just gonna find a way to die in the winter anyway. Possibly that's what's going on. The other thing about it is our relationship's pretty weak. So maybe it's spurious, maybe it's driven by the fact that, you know, in the big hole, we didn't have predators and the calves didn't do too well. So that's a little more uncertain. Oh, yeah, I think I guess maybe I just highlighted these to say, these are ones that are kind of coming out stronger. Definitely, this is kind of the big dog, is that maternal age. And then, you know, kind of variation between maternal females and then the kind of sum total of predators kind of come out strongest. So now Nick could do fun things and rather creative things with trying to see what kind of effect these different things kind of quantify the effect these different things had on population growth. Like I said, we couldn't experimentally, kill all the willow and see what happened, or kill all the wolves, or, infect them all with Coleoptera. So basically what he had to do is he had to take these relationships that were observed between these vital rates and these different factors and say, well, if these relationships hold and we tweak these different factors, how big of an effect would we see on population growth? And you can see the big one is nutritional condition. That seems to be driving a lot of this. And then followed by wolf density, and then Coleoptera. Those are kind of the three that come out. Yeah, let's move on. So I want to kind of focus on the Coleoptera one real quick and just kind of help you understand how he did this. So basically, he took this relationship here and figured, well, we'll say we'll make the cutoff 75% likelihood that they'll die from a health-related issue. So that corresponds to, I think, about 38 infection of 38 worms in the carotid artery. And basically, if you have more than 38 worms, or are likely to, you're going to die. And if you don't, you're going to live. And that's how you kind of get this effect size of saying, okay, we say that, and then we say there's no more Coleoptera to worry about. What would be the bump in the population? The annual growth, you'd expect it to increase by about 2%. This is across all study areas combined. Now, I want to move back to that because, what this means in the big hole is not necessarily what it means statewide. Now, another thing that people are interested in is what would happen if we put the hammer to some of these predator populations. And there's certainly been interest in reducing some of these predators for ungulates in general, but that includes moose. So you could play with this and say, well, if we change the density of these predators, reduce them by 30%, what kind of bump would we expect to see in annual population growth rate? So, it goes from grizzly bears, if those densities were reduced 30%, models would predict we would see a 0.19% bump in annual population growth rate. Wolves would be more substantial, it would be 0.69%. And I think maybe what's jumping out to people right now is like, well, that sure doesn't seem like a lot. And if you look at all of these things, you kind of sit there and go, well, none of these things really seem to be moving the needle a lot, right? And I think that comes down to the fact that, what makes a difference between an animal living or dying, having young? I mean, there's a lot of things that go into that. And so isolating one of these things, it can only have so much in effect. So, I guess, so like one of the things that obviously plays a role is age. That comes up with cow survival, comes up with fecundity, comes up with calf survival. And, there's different age structures across these different study areas. For example, Rocky Mountain Front, a lot of young animals, they're probably going to be doing good for quite a while. The Cabinet Salish area, that has quite a bit of older animals in it. It's really skewed to older age classes there. And so that's going to influence kind of that population growth. And then the big hole looks a little more like the Rocky Mountain Front. But again, these things are interacting with these different variables. And, despite having that younger age structure, there's some of these things that are just hitting the rock or the big hole, both nutritionally and with Elaeophora that seem to be limiting them. So there's some complex interactions there. And that brings me to some follow-up work that we're interested in, not so much a research project, but just kind of putting feelers out there. You know, that Elaeophora effect, I think it was like, what, a 2% increase in population growth rate or something like that. That doesn't seem that much, but that's for all the study areas. In the big hole, we are seeing a lot of these health-related mortalities, very likely playing a much bigger role here. So we kind of want to follow up with some of this, and just some of you are probably familiar, but the thought behind kind of Elaeophora life history was that the definitive host was mule deer, and moose were kind of a dead-end host. And so mule deer would be keeping this on the system, and then, you know, the vector is these horse flies, and then we get into moose and potentially cause problems. That's been complicated recently by some findings in Wyoming, their Fish and Game. It suggests there might be multiple species of Elaeophora. We thought there was one that was, and this was the definitive host. Maybe different ungulates actually have their own Elaeophora. You know, one of the things about the mule deer being the cause of the problem, or contributing to it, is a lot of these places, it's not like it's crawling with mule deer. So you kind of wonder, like, how could the moose be getting this much? There might be other things going on with the Elaeophora species themselves, or with the vector there that could be influencing some of these things. So I think following up, we're trying to get all the samples we can of moose heads to look for Elaeophora, both in the carotid artery, and then if possible, clearing the third eyelid for whatever reason. That's a good place to detect the Elaeophora DNA, and having the lab in Wyoming run those to see what species we might have. I mean, this is really early stages with some of this stuff. And also looking opportunistically at other ungulates. So Vanna and I, mostly Vanna, collected, I think we're up to 12 elk heads out of the big hole to look for Elaeophora in them. We haven't detected it yet, but so this will be kind of some of the monitoring going forward to follow up on these things. And I think that's pretty much what I wanted to share with you. I think it's going for 8:30. Okay. So if there's any questions, I could think.
I just wondered if, you know, the extreme drought, your whole study, the big hole was a drought, right?
I don't know about the whole study, but definitely a portion of it, yeah.
Yeah, I just wonder if that has a big effect on the nutrition that moose are getting in the big hole.
You could think, yeah, you know, I think not so much the amount of forage available, but the quality of the forage, you know, how much digestible and energy and protein is in it, tannin levels, those kind of things. I think it definitely be influenced by it. Well, thanks for your time, folks.
Thank you so much for coming today. Just so you guys remember, we don't meet in December. We'll meet January 15th at Fairmont, but that'll be our annual business meeting, which is typically just for board and staff. And then we'll meet here again on February 19th. And we'll be hearing about the Big Neighbor River Foundation water quality and definitely the bond. And that'll be at 6 o'clock too in February. So, and I won't be here, so Kim will be running the show and doing a great job for that.
Happy New Year, everybody.